extranuclear genome. Right? My voice audible to you? Hello?
Yes, sir.
Right.
Yes, sir.
Right. Mitochondrial genome and the chloroplast genome also completed. That is in third unit, right? Next, to be completed fourth, fifth, sixth in black one. Then what are any remaining in third unit? Third unit. Right. So chloroplast DNA completed and mitochondrial DNA completed in third unit only. One of the important topic is, of course, these two important, chloroplast DNA and mitochondria DNA, they are also important and they are completed. And next 3.17, cytoplasmic male sterility. This is also very important. Cytoplasmic male sterility. Let's see what is cytoplasmic male sterility. Right. First of all, what is fertile and what is sterile? Please respond. What is fertile and what is sterile?
Fertile ante food ichesar, sterile ante em ivvakunde. The
Right.
opposite of this.
Right. Produce. They are produce viable pollen grain. Viable pollen grains that fertile. And sterile means the unable to produce viable pollen. They are unable to produce viable pollen. Cytoplasmic male sterility. What is cytoplasmic? What is cytoplasmic? Where the cytoplasm is located? Hello?
Cell, sir, in the cell.
Right. In the cell, okay, right? Already told you the cell structure. In what are the main components of the cell? Main component. Cell main component.
Endoplasmic.
For example, this is the cell, right?
Yes.
This is the cell. Now tell me the main components. That means cell structure. I'm asking simple question to you. The plant cell structure simply consists of two parts. Respond.
Cell wall.
Right, cell wall. This is cell wall. Okay, outer cell wall, and except the cell wall, remaining portion called as
Plasma membrane.
okay,
Cytoplasm.
okay, yeah, as you said, the plasma membrane, that is the blue color maybe. Okay, this is plasma membrane. And next,
Cytoplasm.
so
Endoplasm and
right, all cell organelles present. That means what is the central position? Central nucleus. Right? Central nucleus. Now, actually, this is cell wall, outer one, next plasma membrane, and next one is the center occupied by nucleus. So, except nucleus, the entire portion, that means from the this region, plasma membrane to nucleus, this region called as cytoplasm. This is called as cytoplasm. Right? Now, cytoplasmic male sterility is the fertile and sterile, the character designated by the nucleus genome, right? Nucleus genome, for example, this is the gamete cell, for example, this is the gamete cell. What are present inside the gamete? What are present in gamete cell? The same common structure. Cytoplasm, nucleus, they are all present. What are present inside the nucleus? Already we completed in first and second unit. Hello. Nucleus components.
DNA, RNA, sir. DNA, sir.
Right, DNA, RNA, they are compacted inside the chromosome.
Chromosomes.
Right, they are the designed as the character, whether it is fertile or sterile. But now, this character designated in cytoplasmic genome. Cytoplasmic genome means what are the cytoplasmic genome? I am asking you a very simple question to you. Other than the nucleus, any cell organelle consists of genome. Hello?
Yes, sir.
Tell me, what are those?
Mitochondria, sir.
Right, mitochondria and chloroplast. Right? Now, this character occurs in mitochondria genome. Let me draw. This is, for example, mitochondria. This is chloroplast. Of course, in mitochondria also have some genomes, DNA genomes, and this chloroplast also DNA genomes. Now, sterility occurs inside these, or mitochondria or chloroplast. Anyone genome shows sterility, that is called cytoplasmic male sterility. That means male means the mainly occurs in male gamete. The sterility, that means the male gametes even fuse with female, not produces, not developed into zygote. So that is sterility. This is called cytoplasmic male sterility. In short form, it is called as CMS. CMS, cytoplasmic male sterility. This is the definition. So do you understand the difference between nuclear genome and mitochondrial genome? Here the sterility mainly occurs in mitochondria or chloroplast genome. That's why this is considered as a cytoplasm. So it's called as cytoplasmic sterility. The simple definition, CMS, cytoplasmic male sterility, is a maternally transmitted from the parental to offspring, maternally transmitted trait whereby a plant is unable to produce viable pollen. It was first reported by Course. The mainly how they are occurs. That means what are the causes to the male sterility? Before that, let's see the male sterility process. Just wait. Male sterility, where they are, just wait. Downloads. Right. Is it clear? Hello? Screen visible to you?
Yes, sir.
Yes, sir.
Right. Let's see the process of cytoplasmic male sterility. We can observe here. First, inbreed cytoplasmic male sterile and inbreed B. First A, B, male fertile, male sterile. Of course, it is male sterile is considered as a female. Is it okay? Hello? Actually, of course, even it is a male one, but the genomes inside the male cell, the so sterile character. That's why it's considered as a female. So the taken the scientist, one is inbreed A, cytoplasmic male sterile, small RRS, sterile. The sterile nature we can observe here. Green color nucleus, this is nucleus, and yellow color are cytoplasm. So sterility in S cytoplasm. Next, cross with inbreed B, line B, first line A and line B. This is male fertile. Male fertile, so F cytoplasm F. When cross with single cross one A and B, the male sterile obtained. Male sterile obtained. That means what? Which character inherited to next generation in this both? Hello? Which character? Line A character or line B character inherited?
Line A character.
Right. Line A character male sterile inherited to next offspring. So that's why this is, so this inheritance already definition clearly said CMS is a maternally transmitted, maternally from mother's parental to the next offspring. So is this fertile or sterile? So sterile, that's why it is called male sterile. Next one, inbreed line C and line D, same cytoplasmic male sterility, and male fertile same as it is. Male fertile, one is male sterile, the same cross, but here the what is the difference in this both? And the first inbreed A, small rr means the character is recessive. Character is recessive, not expressed. Here, this character, of course, it is fertile. Even fused with sterile, fused with fertile, but not exposed to fertile character. Why? Because here the fertile character not a express. It is recessive character, not a dominant, recessive character. That's why the only in this situation the sterility obtained. But second line C and line D, the first as is cytoplasmic male sterility common as in line A. And what about line D? Line D male fertile. Line B also male fertile. Hello? Are you listen?
Yes, sir.
Yes, sir.
Right. So just observe here, in breed B or line B, only small r, that means recessive genes. What is recessive? What is dominant? Hello?
Recessive means it's not expressive character.
That's it. And here in line D, capital R, that means it's dominant. This is dominant character, right? Now cross with fertile and sterile, what would be offspring? Fertile or sterile?
Fertile.
Fertile, because already it is dominant character. That's why the dominant character inherited and male fertile obtained. Then in next generation, when male fertile AB and CD, in this situation, the 50-50% fertile sterile obtained. Fertile sterile obtained. That means, so in this inheritance, sterile character 50% and fertile character 50%. So this is the process of cytoplasmic inheritance. Is it clear? First of all, it is recessive, so sterile, dominant, so fertile. Now next situation, where the small r only recessive character obtained, where the capital R dominate. In the situation of capital R small r, here you can observe here capital R small r. Capital R small r, which character expressed? Dominant, of course, always dominant character expressed. So in this situation, this is the fertile one, the fertile one. fertile one, and this is sterile one. So 50% sterile and 50% fertile obtained. This is cytoplasmic male sterility process. This is very, very important for short answer. Is it clear? Definition and process. Hello? Any doubt in this?
No, sir, please repeat.
Simple. Definition clear? First of all, what is the cytoplasmic male sterility? What is cytoplasmic male sterility? What is sterility? Unable to produce viable pollen. Unable to produce viable pollen. That means inside the pollen, inside the nucleus and cytoplasm, that means mitochondria, chloroplast, anyone, the sterile genes are present. They are continued to the next offspring also. So that's why in first line A, line B, male sterile, male fertile. Of course, it is male fertile but recessive. What is recessive here? The fertile character recessive. In the sense, not express the character. That's why in the offspring also same sterile character obtained. And second, line C and line D, same cytoplasmic male sterile and male fertile. In male fertile, dominant character, taken dominant character. So in this situation, only sterile or fertile obtained in cross 2. Hello?
Sir, listening.
Okay, in cross 2, fertile character obtained or sterile character obtained?
Cross 2 says fertile, sir.
Fertile. What is the reason? Of course, you taken the same
Because
line A, line B, line C, line D, same characters, same genes you taken. Then why the cross 1 fertile, sorry, sterile, and cross 2 fertile?
In class 1, recessive character is there, sir, fertile.
That's it. And cross 2,
Class 2, dominant character is there, fertile. So we
that's why
can
only dominant express the character. Right? Now in double cross, here we can see double cross. Double cross means crossing between cross 1 and cross 2. Cross 2, that means cross 1 male sterile, male fertile, both male sterile, male fertile. In this situation, we can observe the first one, smallest, 50%. That means, just wait, I will clearly explain. Smaller, sterile. Fertile. Right? Do not taken gametes, smaller, capital R, smaller. That means the joint are separated. Gamete. Right? Now cross. One side, this is fertile character. This is sterile character. Right? Now crossing between fertile and sterile. This gamete fused with this one. Which character obtained? Capital R, small r. This is which represent which character? Fertile or sterile?
Fertile.
Right. Now,
Sterile.
tell me the percentage.
50%.
That's
No,
it.
no, no, no.
That's it. Both are 50-50.
Okay.
Why
Okay,
are
sir,
confused?
okay.
Why are you getting confused?
Sorry.
Is it clear now?
Yes, sir. 1:1 ratio.
Of course, that means 50-50. Right? Please respond.
Yes, sir.
Yes, sir.
Right. So let's see the reasons. First reason, mutation. First one, mutation. Due to mutation, the cytoplasmic male sterility occurs. And second reason, environmental factors. Second reason, environmental factors. So they are the main reasons. Then so any what are the benefits of the male sterility, cytoplasmic male sterility? The first benefit is check the self pollination. Of course, it is the sterile one. Sterile one not fused with, that means not produces the fertile seeds. That means
Yes.
not promoted the self pollination also. So it is encouraging cross pollination. That is the first one, first benefit. And second one, facilitate the production of hybrids. How the hybrids are formed? Hybrids are formed by self or cross pollination.
Cross pollination only.
Actually, due to the sterility, completely prevent the self pollination and
Yes.
promote it to cross pollination. Due to the cross pollination, the hybrids it means two different characters are obtained. So hybrid seeds or hybrid plants are produced due to the male sterility, due to the cytoplasmic male sterility. This is about cytoplasmic male sterility. Of course, not clear in our book. I will send the notes later. Right? What I explained now, the same as it is matter, I will send to you. Right. Next one, fourth in it. Structure and organization of eukaryote gene. Structure and organization. Organization already completed. Gene organization completed. Gene organization nothing but you can see here page number 58, 50, 58, 59, 60. Do you have books now?
Yes, sir.
Right. See page number 658 and 59, 60. What are given there? The clearly we can see the nucleosome model and solenoid model already they are completed in first unit.
Yes.
I think no need to explain again.
Yes, sir.
The same matter, that means organization same, genome organization same, carry forward to this. And next one, structure of gene. Let's see the structure of gene. That means 4.4 eukaryotic gene structure at DNA level, clearly given DNA level, right? Already told you the clear difference between gene and DNA. Can you tell the difference between DNA and gene?
The functional unit of the DNA is the gene.
Right, that's it. The functional unit of DNA called gene. Right? So mainly the gene structurally made up of, just wait. Right. This is the gene structure. First one is DNA. Are you able to see the screen diagram?
Yes,
Hello?
sir. Yes, sir.
Yes, sir.
In first diagram, that is a DNA with gene. That means this is DNA. This is this entire one, 5 and 2, 3, and for example, this 1 and 2, another 5, 2, 3, and that is completely called as DNA. But where the red color patches, only red color patches, that is the active site. Only red color patches called as gene.
Yes.
This is gene, this is gene, this is gene. But the entire length is called DNA, right?
Yes, sir.
Entire length called DNA, right? Now I picked up only gene structure. We want gene. What is the word? The topic, gene structure. Let's see the gene structure. If you take the gene, the clearly it again separated into active, inactive sites, such as exons and introns. This one colored one, exons, active and functional, non-functional, colorless. Non-functional, colorless. These are called introns. These are called introns. So the introns should be removed. I will tell you how they are removed later. Okay. So this is gene structure. So mainly gene, functional exons made up of introns. That is the first one. And second one, apart from that, in gene, another structure, promoter and operator structure and structural part also present inside the gene. Promoter, operator and structural. Just wait. Is it clear? Diagram.
Yes, sir.
Have you drawn? Have you drawn
Yes,
this
sir.
diagram? Okay.
Yes.
Right. In clear structure, of course, first diagram, first only in gene. Just wait. Exon, intron, exon. Already told you intron should be removed, but only the functional is exons. That's why I take an exon structure. If you take exon structure, this structure appear like this. That means this exon, this exon region, promoter region and operator region and structural region. This is structural. Be careful. This is very, very important. If you understand DNA or gene structure, you can easily understand the further units. Further units such as unit 4.
Restriction enzyme, endo.
Sorry, unit
like
5,
that.
gene
Yes, sir.
expression in eukaryotes and
Okay.
regulation gene expression in eukaryotes. Gene expression, gene regulation. These both are will be easy if you understand the gene structure. This is very, very important, right? So once again I tell you, first DNA. In DNA only active part gene. In gene only the functional group exons. In exons, now the further divide into promoters. This is promoter, operator next, and structural. This is this coordination of these three group of genes. This not a genes, group of subgenes, promoter subgene, operator subgene, and structural subgene. The cooperation of these three, the all functions are carried out. All function means DNA synthesis, DNA replication, and protein synthesis. They are all carried out with help of the regulation of promoter and operator and structural genes, right? And I will tell you, I will explain clearly in further units, right? Is it okay now? Hello?
Okay, sir.
Yes, sir.
right? Clear? Right. Now,
बड़ा बात करती मैं
don't talk, please.
सॉरी
Right. This is this complete DNA. This blue color gene, complete gene. This is an exon. This is an exon. This is introns. Colorless region introns, right? So now, right, in our book given clearly, now you read page number 61. Size of eukaryotic genes can vary greatly. Size, that is the common. That means it's not a uniform gene structure. In an organism, the genes never uniform in the matter of sizes. That's why the sizes, they are variations. That means from 1 kb stones to as much as 2500 kb. For the dystrophin gene. What is dystrophin gene? Have you heard this word? Dystrophin.
नो सर
Dystrophin is the largest gene. Dystrophin is the largest gene in human being, located short arm of the X chromosome. The function is connecting nuclear muscle cells, connecting
Okay.
muscle cells. That means actually genes. That means of course each and every cell, each and every tissue made up of cells. Inside the cell nucleus, inside the nucleus and chromosome genes are present. The genes, number of genes shows the variations, shows the variation. For example, dystrophin genes, the highest number, highest weight to 2500 kb in X chromosomes, in X chromosome only, right? So in typical eukaryote, the region of the DNA coding protein usually not continuation. This is very important. Of course, already I told you, the gene not a uniform, of course, it's made up of exons and introns. Here, the what we given non-coding sequence not does not involved in protein synthesis. So this is, for example, this is exons, this is introns. Already told you these exons only functional.
यस सर
Exons only functional means exons only prepare the protein, can prepare the DNA strand, but not the introns. The introns should be removed, right? Introns should be removed. Introns should be removed.
But what is the function of them, sir? Is there any reason that the introns are present?
Introns no reason, because the introns itself unfunctional. The introns only involved in packing of genes, like a nucleosome, like a histone proteins. Do you know histone protein?
Yes,
Hello?
sir.
The histone
Histone.
protein, the useful to compactly arrangement of the genes. The compact arrangement of DNA strands or genes. In such a way that introns also same function. The only, that means the function means only involved in packing, but not in protein synthesis, but not in DNA synthesis. because it is non-coding, does not code any character, non-codings. So these non-codings are called exons. Non-coding parts called as introns.
Introns.
Right. Coding part called as
Exons.
exons. Coding one exon, non-coding intron. Right?
Yes.
Clear?
Hmm.
Right. So just wait. Now introns should be removed. That means after remove, I will tell you how they are removed. After remove of these introns, where the introns are present, they are removed further during transcription process. During transcription process, I will tell you clearly in protein synthesis. Okay? But after removal of the introns, the structure would be that's it. That means join the only functional group. This is exon. This is exons. Though only exons are joined after removing of the introns. After remove of the introns, this is called splicing. This process called splicing. This process called splicing. That means removal of introns. Removal of introns called as splicing. Now, right, let's see. Intron sequence contains some common feature. Most of the introns begin with GT in this region. Begins with sequence guanine, thymine, or guanine uracil. End with AG. Right. The over 90% of the sequence between 5' to 3', the mRNA is introns. This is 5', this is 3'. 90% non-functional, 90% only introns. Right. Where the GT guanine thymine starting, ending adenine guanine. These two bonds break from the exons. As a result, it is removed. I will explain clearly how the spliceosome process occurs. Right? So just you remember from the guanine thymine and adenine join starting of introns, guanine. It breaks from the exon nucleotides. In the same way, ending point guanine break with exons nucleotide. Now it should be removed. These both exons are joined. Right? So this is only functional genes. Right? In functional genes also, again, the two boxes are present. One is Tata box and CCAET box. That means first one, exons, only in exons. Adult told you, promoter, operator, structural present. Promoter, operator, structural. These all together combined form an exon. Now let's see promoter sequence. Now let's see only promoter region, right? This is the gene in a order. We are observing the gene order. But first one, DNA with gene, exons, introns, only exons. In exons, promoter, operator, structural. Now see only promoter region only. Only promoter region. Hello? Are you follow me?
Mm.
Yes, sir. Yes.
Structure clear or not?
So
Yes, sir.
actually, we are having the nitrogen bases are the triads now. Then why we are taking only GU, GT, AG like that?
Sorry?
So actually, the nitrogen bases we express in terms of triads.
Yes, okay.
But here, why we are writing GU, GT, AG? That I didn't
Okay,
understand.
okay. What are the nitrogen bases?
Adenine, thymine, guanine, and cytosine. Uracil is there, but
Right.
that's
What
not
I
there in
said
DNA.
now?
Triad.
What I said, the first starting with introns, ending with introns.
Okay. Yes.
What I said?
GU, GT, something like
What is
that.
GT means?
No.
What
Guanine
is
and
GT?
thymine. Guanine and thymine.
Right. Is there is there not a nitrogen basis?
It's not a triad, na.
Triad.
It's not a triad. When we write DNA structure or RNA structure, AUG like that, we used to write in terms of
Not a
triad.
triad. They are triplet codons.
Yes.
Triplet codons
Triads are
expressed
triplet
on
codons.
mRNA.
Yes, it is also, I think, mRNA that is involving in protein synthesis, na, sir?
That's why I'm telling you.
Okay.
I will clearly explain in protein synthesis. That is next in it.
Okay.
Protein synthesis next in it.
Yes, sir.
I will explain
Okay.
clearly what are the triplet codons. What is the starting codon? What is the ending codon? Just you listen here. As you said, the DNA is made up of nitrogen bases, right?
Yes, sir.
That's what is shown in this diagram. Nitrogen bases. The nitrogen base is guanine, is nitrogen base. Thymine is nitrogen base. Adenine, nitrogen base. Guanine, nitrogen base.
Nitrogen base, yes.
Right?
Okay.
Of course, you just assume they are in triplet.
Okay.
They are in triplet. But this is not a protein synthesis.
Okay.
This is not a protein synthesis. If it is protein synthesis, I will clearly explain.
Okay.
In protein, what is protein synthesis?
Synthesis of amino acids and by which results the proteins? Amino acid synthesis.
Right. How the amino acids are synthesized?
On mRNA.
Then how the mRNA synthesized?
Which is releases from the ribosomal RNA after replication, some transcription or some transcription process will be there.
Right. Just wait. What is transcription?
And one of the strands of DNA.
Enough.
Another.
What is stand for DNA?
It works as a template and it produces a similar copy of it, which is having uracil instead of thymine over there.
No need at all. Only one I want question. The template is DNA, right?
Yes, sir, DNA template.
Strand, RNA strand is produced.
Right. On the DNA strand, mRNA strand produced.
Yes,
that is transcription, right?
Yes.
So before that, you should know about DNA structure.
Yes.
Without DNA structure, how will you understand about the protein synthesis?
Yes, I do agree, sir.
That's why I am clearly explain about the DNA structure. What are inside the DNA, gene, exon,
inside the
promoter.
gene, exons, introns,
If
and
you
in exons.
know about promoter, operator, structural, they are all involved in protein synthesis later.
Okay.
Already tell you in the beginning. If you clearly understand about the gene structure, The transcription process translation means protein synthesis would be easy to you,
Okay.
right?
Okay.
Right.
Thank you.
Next. Now promoter sequence. Promoter sequence. We started with DNA, gene, exon, promoter.
Promoters.
Now, right? Let's see the promoter
Promoter.
structure. What are the components of promoters? Let's see. Can I rough this?
Okay, sir.
Lalita, tell me. Why are you raising your hand? Any doubt? Lalita, is there?
No, she left, sir.
Okay. Right. Right. This is promoter sequence, only red lines. Promoter left side upstream. This is important. These words very, very important. Listen. Where is the upstream and where is the downstream? They are useful in protein synthesis, transcription, translation. So this is red
Okay.
lines. Red lines.
Promoter sequences.
The red stripes, promoter region. Left side upstream and right side downstream. This is Tata box. So in promoter regions, just side the promoter region, just side the promoter, this yellow line, TA,
Tata box.
TA. This is called consequence sequences. If you read this direction Tata and this, sorry, TA, that is one end, 5 end to 3 end. The same, the reverse process, TA. This box called Tata box. This is the initiation site of the protein synthesis. That means, first, what is the first stage of protein synthesis? Transcription.
Yes.
Transcription started with Tata box. This
Yes,
initiation
sir.
site. Tata box is the initiation site. This is the closely arranged to the promoter site, promoter region. Actually, this entire portion this entire one called as promoter region. This entire one called as promoter region. In promoter region, first promoter sequence and just side Tata box, and this end upstream side, this upstream side, this up not given here actually. The upstream side, the silver line upstream enhancer, the place codings are CCAT. So this is the order sequence. This is the order sequence CCAT or cat box. CCAT or cat box. I will explain clearly what is the function of Tata box and what is the function of CCAT box in transcription. Okay. Just
Okay, sir.
here you can focus on the gene structure, what are present, what are the components of gene. That's it. Okay. Is it clear now?
Yes, sir.
Right.
Okay, sir.
You can see first one. Complete structure. Complete DNA. This is, for example, DNA. First 5 and 2, ending 3. What are present in DNA? DNA enhancer region or promoter, this promoter region, this one. Promoter region, exons, introns, they are all present. A transcription process, due to transcription process, from the DNA, what is transcription? From the DNA, mRNA strand prepared. This is
Yes.
DNA strand, and next mRNA prepared. Of course, you can assume here always triplet codons are present. Triplet. This triplet, always triplet mRNA. That means the first formed mRNA, exons are present and also introns also present. Exons in the form of triplet codons, genes in the form of triplet codon, and introns common without any code, non-codons. So they are present. But later, due to the RNA processing, these introns, this white color, these introns are removed. Now last diagram, only pink color, only light pink color. So this is only exons. Only exon, this first one start codon, ending stop codon. Here, ending stop codon, right? So this is gene structure, and clear gene structure I will explain in during transcription process, right? Is it clear? Hello?
Okay, sir. Okay.
Right. Now, in genes, alternatives start and stop producing related proteins.
Okay.
That means if you take genes, two genes clearly present. One is start codon, for example, another one is stop codon. GAA, example GAA. This is stop codon. One is start and one is stop codon. This is another structure of gene. That means start codon, stop codon. Now overlapping genes. This is also important. Overlapping genes. Overlapping genes. Fatima. Hello? Maharush Fatima. Any doubt? Hello?
Maybe mistakenly she has done.
Okay, no problem. Right. Overlapping genes. Overlapping is also part of the genes, part of the DNA. Overlapping genes. Right. And overlapping genes also called dual coding genes. Overlapping. What is overlapping first in general? Hello?
Overlap.
I'm asking general question, but not related to genes.
ఒకదాని మీద
One is covering something else.
That's it. That's enough. Okay, right. So it is also called dual coding genes of DNA or RNA. The same where it is repeated one above another. That is overlapping. If you understand the overlapping genes, you can understand about two different frames here: reading frame and open reading frame. First reading frame, this is not overlapped, right? Codons, 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 codons. The same way open reading frame means this is reading frame. Open reading frame means should have start codon and stop codon. Start and stop codon. That is open reading frame. Open reading frame. Same reading frame without stop and start. With start codon and stop codon, that is called open reading frame. Then here overlapping means in overlapping in a homologous chromosome, two genes. That means, for example, this is one chromosome genes and this is another chromosome genes, right? Tell me some sequence. Start for example is AUG. Here also
AUG.
you tell me some sequence of codons.
CAA.
A,
CAG.
C, okay. Next.
T nahi.
Oh,
C
this is
kaadu.
uracil. Uracil. Yes, it is RNA, uracil. Okay, next.
ACG.
ACG.
GAA.
GAA. Okay. GAA. Right. This sequence now. Next sequence here. Tell me some another. Anyone? GUC, GCA, and GAA. Here also same GAA. Here GAA. Now this is called overlapping. That means same sequence.
Okay.
Same sequence. Just assume here if they are if these both involved in protein synthesis, can they provide same protein or different protein?
That depends upon the earlier former sequence, na, sir.
Just you can see here the sequence. Also, you tell me.
They are same expression.
How can it same?
GAA is same, na, sir.
Of course, this both are starting codon same. No change at all.
Huh.
Then what about
The
second?
protein is different, sir. AUCACG. That is different codons are there, and the second one also different codons are there.
That means
So the
these
proteins
AEC
are
code for just wait. AEC code for same amino acid. GEC code for same amino acid.
no,
Then
No.
how they are synthesized the same proteins?
no, different proteins only. So they produce different proteins and different amino acids.
That's it. They produce the different proteins. Different proteins. Of course, they are the different protein. Open that means it should consist open reading frame. Different open reading frame. This is different open reading, and this is also different open reading. That's why this synthesized two different proteins. Synthesized two different proteins. So as it that means this is due to the overlapping. This is the overlapping. Of course, the same sequence here. GAA same sequence. Here also same sequence, but the synthesize the protein different. Synthesize the proteins different. So this is overlapping, and this is first discovered in 5 genome 5174 bacteriophage 5174 bacteriophage first. So this is overlapping. That means overlapping means first one you should understand open reading frame. Here two different open reading frame. Individually this prepare one protein synthesis and this also one protein strand. As it is overlapping, that means the same sequence repeated. Of course, they are the repeated same sequence, although they produce the two different proteins. So this is overlapping of genes. Next one, alternative splicing. Second one, alternative splicing. What is splicing? Already told you. Splicing means removal of introns. Removal of introns. Right?
Yes, sir.
We can see clearly in diagram 1, in page number 64. Page number 64, just wait. Page number 64. Splicing type. First one. Blue color. Blue color. Blue color.
Exons.
Yellow color. This is gene. Now yellow color is exon. Sorry, yellow color is intron. Yellow color introns. Right. Splicing means removal of introns, right? Here
Yes, sir.
alternating splicing. Alternating means this two introns. Skip the exon. Intron. Sorry.
One exon followed by intron.
Exon. Right? So this common. So introns removed, the exon joined. As a result, exon region. This is one type of alternative. And second one, exons, introns, in this situation, exon skip the one introns. Sorry. exon. This is also exon. This is intron. Exon. Actually, instead of skip the introns, in this situation, the exon skip another exon and join with introns and exon. So in this situation, one exon form, this exon skipping, removed, and next one. What is joined here after exon?
Exon only. Intron.
Intron.
After exon.
And again
Exon.
exons. And again exons. For example, 1, 2, 3, 4. 1, 2, 3, 4. These numbers are exons. Exon, 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 exon. There you can you understand one exon removed. One exon removed. Either 3 or 2, anyone removed. That means after splicing, in this situation, 1 common, 2 common, 1 removed. Already told you 3, for example, 3 removed should be 4. That is one end. Or the first, maybe first one removed should be 2, 3, 4. Or second one removed should be 1, 3, 4. So this is exon shuffling. This is called exon shuffling. That is shuffled. Skip the exons. Sometimes some of the exons also skip and joined with introns instead of exons. So this is one of the
Mutation.
splicing process. Actually, we know the splicing means. What is the general definition of splicing?
Removal of the introns.
Right, but sometimes removal of exons also may occur. That is called shuffling. That is called shuffling also may occur. That is here called exon shuffling or alternative splicing or exon shuffling. Is it clear? Hello? First
Yes,
one,
sir.
splicing definition. Splicing, remember, splicing means only removal of introns. But sometimes may also the removal of exons also may occur. This is called exon shuffling or alternative splicing. Right? Next
Yes,
one,
sir.
gene family not clear. I will explain clearly.
So this shuffling of this splicing only leads to the mutations.
Yes,
Is
of
it
course,
responsible?
200%. If the one of the active site is removed,
Yes.
that means, for example,
One of the function is going to be affected.
that's it. So for clear understand, for example, this is actual sequence 1, 2, 3, 4.
Understood, sir.
Right? Clear? No need to explain?
No.
Okay. Repetitive DNA. Actually, gene family, repetitive DNA, organization gene. Okay. Okay. Right. This rest of the portion I will complete later. That is gene families and repetitive DNA, because they are to be prepared notes. I think it's not clear in book. I will clearly prepare the notes. After that, I will explain. So both this repetitive DNA and also gene families and organization of eukaryotic gene. Organization of eukaryotic gene clear, but not in order. Organization of eukaryotic gene also, the promoter, enhancer, they are given, but not in a order. I will arrange the correct order. After that, I will explain. And you can observe here downstream and upstream. Tata box, they are already repeated. Tata box, regulatory elements, promoter sequence, structural genes. Actually, the organization of eukaryotic genes, I think I thought actually it would be completed during explanation of protein synthesis. The time of protein synthesis, they are again repeated. And what is enhancer? What is promoter? What is Tata box? What is CACT box? I will explain later clearly during protein synthesis.
Okay.
Okay.
Okay, sir.
Right, transcription. Translation. Where is transcription? RNA chain elongation, termination. Okay. Eukaryotic translation. Okay. First, now I will explain only the concept of DNA replication, the transcription and translation. After that, I will clearly explain later, right? Okay. We know clearly about gene structure.
Till now we have seen the gene structure, which is having the exons and introns,
Right.
where the exons are the functional units, where the introns are non-functional parts.
Right. Now gene expression in eukaryote. Gene expression. For example, in rough gene. Express the character. So the gene express the character at the certain time only, isn't it? Hello?
Yes,
For
yes.
example,
The hormones regarding to.
of course, but for example, the leaflets, that means plantlets are emerged out from the seed. When the plantlets emerge out from the seeds, immediately they produce the flower.
Cotyledons produced.
Cotyledons. Later they developed as a
Shoot system,
shoot, root,
primary,
leaf.
yes.
That only primary plant body develop. Then what about flower?
Afterwards. After
Afterwards.
certain time.
Where the flowering gene?
Within the seed itself, it was under dormancy. It is not expressed this phenomena. Of
Why it is not
phenotype.
expressed?
There
It
is
was under some
there is no need
So-
to express at that time.
No need to express.
somewhat dormancy like condition.
Okay.
Repression.
What is the reason for depression?
Repression. For the expression of the gene, it needs certain conditions. Plant should be there, then only it can able to produce the flower. When there is no proper formation of the plant structure, then the nutrients cannot be supplied, enough nutrients cannot be supplied for the flower, which in turn result to the fertilization, fruit formation, seed formation, etc. So for that, the environment should be created for the plant to get produce the required flowering genes and all in the later on period.
Okay. So I to provide sufficient nutrient and sufficient weather, sufficient environment to the simple plantlets. Can it produce flower? I will provide all.
Maybe.
What you said, I will provide all. Approximate temperature, environment, nutrients. They are all provided. As you said, they are all provided. I will provide to the plant. Then I will produce the flower immediately.
Climatic conditions also required. No?
Of course, the climatic factors also provide.
Plant body structure should be there, distinct structure.
What you said, they are all I will provide. That means simple. Even if you provide all nutritions, all sufficient environment, optimum temperature, even if you provide all, the plant.
The genes need certain
produce.
time to express their phenomena.
Let me complete.
Phenotype.
Let me complete. Even if you produce what you said, they are all nutritions, all temperature, sufficient environment, the plant not provide flower initially. That means immediately, as soon as emerged, not provide flower. But take time. Take time for providing flower. The meantime, where is the flowering gene? Where is the flowering
genes
gene?
are arranged in a sequence. First, which gene is supposed to express is phenotype, that express the phenotype. Later on, when the turn of this flower controlling gene time, the sequence will be there on the DNA. So when the time of this expression of this controlling gene, then only it expresses the phenotype.
That's it. Controlling. You said only one word, that is controlling gene. Of course, gene is included inside the cell, but not expressed. It is controlled. It is controlled. Gene is controlled. Right? When it is expressed, then when it is expressed. For example,
when the
gene.
exons are
Right. For example, gene. Through the transcription process, mRNA prepared. Transcription. For example, this is the gene. With introns exons also present. Introns exons also present. Now, unmatured mRNA, first unmatured mRNA from unmatured mRNA means introns present, exon present. Introns, exons present. That is unmatured mRNA. Right? Later, introns are removed. Only exons are formed. Only exons are formed. Now it is fully matured mRNA. Fully matured mRNA. Later, this starting region, I will tell you later. Capping tail, adenine tail formed. I will tell you later what is the reason why the cap is formed, how they are formed, how the tail is formed. I will tell you that. This is for concept development. Right now, anyhow, the mRNA prepared from the gene. The related to character gene. I am explaining about flowering gene. This is flowering gene. To produce the character of flower, these all aspects are one by one takes place. All aspect. That means from the gene to mRNA prepared. Immature mRNA. After removal of introns, matured mRNA prepared. After development of matured mRNA, though this matured mRNA, first of all, this transcription location, where the transcription takes place. Hello?
on DNA.
Of course, in DNA. Where is the DNA? In eukaryotes, where is the DNA?
Nucleus.
In nucleus. That means the transcription process occurred in nucleus. After formation, mRNA
Yes,
need to be transported to cytoplasm. Right?
Yes, sir.
Now, Inside the nucleus, mRNA transported to the cytoplasm. Now, where the mRNA stand? mRNA present in cytoplasm, right?
Yes.
Okay, what are present in cytoplasm?
The cell organelles.
All cell organelles and also amino acids also present.
Yes.
And tRNA also present.
Yes.
tRNA, rRNA,
Yes.
amino acids, all cell organelles except the nucleus, remaining all cell organelles present. When the mRNA reaches the cytoplasm, immediately transfer RNA, tRNA active and attached with proper amino acid. Of course, here 20 type of amino acid present in cytoplasm, 20 types. For example, here the one sequence AUG sequence. AUG sequence only code for tRNA, one type of amino acid, that is the for our better understanding amino acid 1. AUG. These AUG only code for amino acid 1. Now it is attached with tRNA and brought to this region. First one amino acid clear. Second sequence, for example, GCA. And GCA code for another tRNA, another amino acid. That amino acid should be, for example, amino acid 2. Now amino acid 2 bind with tRNA and brought to the GCA region. It would be continued. Hello?
Yes, sir.
Hello? What I'm saying, AUG triplet codon
Codes for one
tRNA
amino acid.
brought only one, not another. Only AA1 only code for AUG, not GCA. GCA only code for amino acid 2 amino acid, but not AUG. Right? Like this, another triplet codon, here another triplet codon until the nonsense codon, until the end. So like this, different type of amino acids are brought towards triplet codon and it synthesizes the group of amino acid, right? Group of amino acid. So for example, this is AA1, 2 amino acid, 3 amino acid, like this, 1, A2, A3, A4, A5, A6, A7. Like this, number of amino acids all together form a peptide bonds and developed into chain, like this chain. Now this is called one amino acid, another amino acid bond with peptide, bond with peptide, bond with peptide. Now this is how many peptide bonds here? How many peptide bonds? How many peptide bonds? Each and every amino, between each and every amino acid, one peptide bond present.
Yes.
Then here how many? 1, 2, 3, more than one?
Yes, sir. 1, 2, 3, 4, 5, 6. So many
That means
peptide
more than
bonds are
one.
there.
Of course, here are given, of course, 10, 9, but the more than one peptide bonds are present.
Yes.
So this is called, more than one called as poly. This is called as polypeptide. This polypeptide chain called as protein. Now protein synthesized, right?
Yes, sir.
Protein synthesized. Proteins chemically enzymes. Protein nothing but enzymes, right?
Some of the proteins.
No, here actually these enzymes are chemically proteins.
All the enzymes are chemically proteins.
Right, that's what I'm saying. Enzymes, chemically proteins. Now these proteins act as enzymes. Now this enzyme acts on flowering substrate. Flowering substrate. Flowering substrate. Flowering substrate present initially. flowering gene also present.
Yes.
But this gene not active until produce the flower. When the time reached to produce flower, these all aspects are one by one completed. mRNA prepared, amino acids group prepared,
Polypeptide
amino
chain
acid
prepared.
bond with peptide bonds. The peptide bonds all together form a protein.
Protein.
Now this protein enzymes. Now this protein enzymes already prepared, and here the ready flowering substrate present. React on flowering substrate. Now only the flower produced. Then flower produced. Is it clear the concept of protein synthesis? Hello? Concept of protein synthesis and gene expression, gene regulation also. Hello?
Yes, sir.
Is it clear or any doubt?
No, sir, we have understood. Understood, sir. Personally. We can't able to hear you.
Clear the concept of gene regulation and expression. That means
Yes.
protein synthesis process and how they express the character.
When and how they express.
Please respond.
Yes, sir. When and how they express also.
That's what I am asking you.
We have understood when and how they express, sir. We have understood. I have understood.
Okay. That's what. Then what about others? Please respond, all of you. If you understand, say yes. Or say no.
sir.
Yes, sir.
Yes, sir.
Understood, sir.
Right. If you understand about the concept of this gene structure and the gene protein synthesis process and the character expression process, only this concept, if you understand this concept, you can understand easily about further classes. Right? Okay? Right. So now, tomorrow I will continue this session. That means only the protein synthesis part. Now I want to complete about tissue culture portion. Tissue culture.
Okay, sir.
Tissue culture and tissue culture application. Right?
Okay.
Page number biotechnology. Right.
I don't have the book.
Page number 193. Biotechnology part. It may be boring. The same topic will be continued. May be boring. That's why change the topic. Now biotechnology.
Not boring,
Technology.
sir. It is plays an important role in understanding the how the plants and all expressing the
Of course.
phenotype.
Right. Anyway.
Let's discuss tissue culture.
Right. Biotechnology. What is biotechnology?
Biology applications of the biotechnology with the help of so many other subjects like maybe the biostats or the mathematics or the chemistry mingle with other subjects and explained and studied in a broad manner.
That's it.
No, it's a lot.
Biotechnology. What is technology? Hello? What is tech? I am asking very, very simple. Of course, they are not a book definition. They are all general definition. Technology.
Techniques
Biotechnology. What is first of what is biology? Biology.
Study of life, sir.
Study of life. What are technology in life?
Using very
Application of
scientific techniques.
and the scientific knowledge to the living organisms to produce
Scientific innovations. For example,
Microscope, like this, sir.
oh, of course. In early stages, the scientists only observe the plants, how they are produced, one generation to another generation. It takes time. For example, mango tree, which provide fruit from take some 10 or 20 years. And for example, neem tree, rice tree. One example is mango to take more years to produce the flower. But now due to the technology, the mango tree in a crop field, how many years taken to produce the fruits? After planting a plant, it takes more than 10 years.
Earlier, yes. Now it is
20 years or 10 years.
earlier it used to take much time, but now
I am asking, in conventional mango tree, it takes more than 15 years approximately to take. Actually, if you seedling mango seed, planting mango seed, it germinate, provide plantlets.
More time, it takes more time to
Provide
germinate.
plantlets and develop in primary plant body and provide flower and fruit. It is time taking.
Yes,
Now,
sir.
due to the technology, within 2 to 3 years.
Yes,
2 to 3
within
years.
2 to 3 years, we are getting fastly that fruits, sir.
That is one of the technology aspect. And another
Yes.
one, actually, we must have seen some of the important crop plants can easily infected with pathogens, right?
yes.
Can you give any example? So many example,
This
anyone?
mirchi, any crop like mirchi or else paddy.
1 minute.
Groundnut.
Just wait.